Hello and welcome to our continuation on the topic the cell and its environment. We'll be looking at hemolysis and crenation. The word hemolysis is a combination of two words, hemo and lysis. Lysis means to split, while hemo was gotten from the hemoglobin pigment which is contained in red blood cells. Therefore, hemolysis can be said to be the splitting of the red blood cells. Hemolysis is the splitting of the red blood cells. And this splitting is due to excessive intake of water. That is excessive absorption of water. Normally, the blood in your body consists of four components: the blood plasma, which is the liquid portion of the blood, the red blood cells, the white blood cells, and the platelets. Now, if the red blood cells in the blood absorb excessive water, there will be what? Lysis. This will result in what? Bursting of the red blood cells. And that will bring us to the definition of hemolysis. Hemolysis can therefore be referred to the bursting of red blood cells in the plasma due to excessive intake of water, or we can say due to excessive absorption of water. This excessive absorption of water will result to what we call a high sugar pressure, that is a high turgidity. And because the red blood cell is an animal cell and not a plant cell, it lacks a cell wall, and because it lacks a cell wall, it cannot resist the elastic nature of the sugar pressure. This will result in the cell lysing. It is important for you to know that the normal blood plasma and the red blood corpuscles are isotonic. When we say isotonic, we mean that they have the same osmotic concentration. That is, both the red blood cell and the plasma have the same osmotic concentration. If, for example, the osmotic concentration in the red blood cell is 5%, that in the plasma will also be 5%. Now, if for some reasons the concentration of salt in the plasma falls, that means the plasma will become what? Hypotonic. Because based on our teaching in osmosis, we said that the concentration of a solution that falls is hypotonic to the other word solution. In this case, the red blood cell is said to be what? Hypertonic. That is, the tonicity or the concentration of salt in the red blood cell is higher as compared to the plasma. In such situation, water will enter into the red blood cell by the process of osmosis through the cell membrane. And since water is entering into the red blood cell, we say this is endoosmosis. And you should remember that in osmosis, the water is coming from the hypotonic solution and it enters into the what? Hypertonic solution. That is why there is absorption of water by the red blood cells. Continuous absorption of water into the cell will make the cells very turgid, and subsequently the cell, that is the red blood cell, will what? Eventually burst. I will be showing you a diagram to fully illustrate this concept. The diagram you can see shows the process of hemolysis. The first diagram there shows the normal red blood cells. This is the normal red blood corpuscles. You can see how it looks like. Now, due to the hypotonic nature of plasma, or when you put this red blood cell in a more hypotonic solution, let's say distilled water, the red blood cell is going to absorb this water, and when it absorbs it, it swells, and this will result to the next diagram you will see. That is the result of the red blood cells absorbing what? Water. Subsequently, because there is no cell wall and just a cell membrane, the red blood cell will not be able to withstand the turgor pressure that results due to the absorption of water, and this will result to the lysis of the membrane. That is what you can see there, that there is what? Lysis or rupture. The red blood cell subsequently what? Burst and releases what? Is content. This briefly explained the process of hemolysis. The consequences of this hemolysis is that hemolysis will lead to anemia. And what is anemia? Anemia is a condition in which the total number of red blood cells which are normal are reduced. Anemia, if ignored, may eventually what? Result in what? Death. And like I have mentioned before, and I want you to remember this and put this at the back of your mind. The reason why there's hemolysis in the red blood cells is because animal cells have no cellulose cell wall. And because of lack of cellulose cell wall, they can resist the turgidity caused as a result of absorption of the water during hemolysis, and hence the bursting of the red blood cells. Now, on the other hand, what if the concentration of the plasma is higher than that of the red blood cell? What happens? From our definition of osmosis also, we see that the water will move from the less concentrated solution, which is the hypotonic solution, to the more concentrated solution, which is the hypertonic solution. If the plasma has a higher tonicity, that is hypertonic or more concentration than the blood cells, this means that water will leave the red blood cells by the process of osmosis too. And this process is known as exoosmosis, since it is leaving the red blood cells. And as a result of this, the red blood cell is going to shrink. The shrinking of the red blood cell is what we call crenation. I'll be showing you a diagram of a crenated red blood cells. That is the diagram of what? A crenated red blood cells. What has happened here is that the red blood cells has lost what? Water. You can see the structure of the red blood cells here. They are deformed. These are various red blood cells that have been created. They have lost water, and hence the structure you can see on the screen. To summarize what I have said thus far, when a red blood cell is in an isotonic solution, there is zero osmosis. When a red blood cell is in a hypotonic solution, there is hemolysis, that is bursting of the cell. And when a red blood cell is in hypertonic solution, there is crenation, that is shrinkage of the cell. Now, in what condition can there be hemolysis? I will list some conditions in which we can see hemolysis. Number one, when there is low concentration of solutes in the blood plasma. Two, when there is high concentration of solutes in the red blood cell cytoplasm. And three, when there is defect in the red blood cell membrane. And in what condition can there be crenation? Number one, when there is high concentration of solutes in the blood plasma. For example, in the frequent use of laxative. And two, when there is low concentration of solute in the red blood cell cytoplasm. This brings us to the end of hemolysis and crenation. We are going to be taking some questions now. Our first question goes thus: a major reason why the red blood cell can survive in the blood is that A, blood plasma and the red blood cells are isotonic. B, red blood cells are hypotonic to the plasma. C, the red blood cells are hypertonic to the plasma. And D, the red blood cell receive digested nutrients. If you have been following the lecture we have thus far, you will understand that the answer will be A. And this is because that red blood cell will survive only in the blood if the blood plasma and the red blood cells are isotonic. That is, the solute concentration of the red blood cell cytoplasm and that of the blood plasma are the same. But in a situation where the solute concentration in the plasma of the red blood cell is higher than that of the blood plasma, the red blood cell will absorb water from the plasma and swell. And because the red blood cell does not have a cellulose cell wall, a point will come where it will finally burst, and this we call hemolysis. But when the solute concentration of the cytoplasm of the red blood cell is lesser than that of the plasma, the red blood cell will shrink, and this process we said is a crenation. Now, to our next question. It says hemolysis occurs when red blood cells A and blood plasma are isotonic, B are hypotonic and blood plasma hypertonic, C are hypertonic and blood plasma hypotonic, D are weaker in concentration than blood plasma. From our lectures and from the teaching we just had, you will know and understand that hemolysis occurs when the blood cells are hypertonic, that is at a higher concentration, and the blood plasma are hypotonic at a lower concentration. This will bring our answer to C, are hypertonic and blood plasma hypotonic. So our next question: It says, mention 3 conditions that may cause hemolysis. From what we learned thus far, we see that the conditions that can cause hemolysis include: number 1, low concentration of solute in the blood plasma. 2, high concentration of solute in the red blood cell cytoplasm. 3, defect in the red blood cell membrane. We have come to the end of our question and answer series. Now, I'll be showing you some exercise for you to try your hands on. This exercise were taken from our textbook on hidden facts in biology, page 68 through page 69. Try your hands on them for a better understanding. You can get the answers at the back of the book. Thank you for listening.